something like this. Once I share my screen with you in 3, 2, 1, boom. Wow, man, was that good. Adam, aren't you impressed with that? I know I am. Your DNA notes look just like this. Now, I know that you're thinking to yourself, Self, why did I do this yesterday if he's doing it today? Because I want to make sure that everybody gets this information today. That's going to be the important part. And so that we've got because this unit in particular from today, or technically yesterday, to today, builds to tomorrow, and then Thursday and Friday. And if you don't understand today, we're going to have a harder time with the rest of the week. Now, the other thing is this. If you taking notes today, I'm going to be taking a picture of all the notes and sending it to Mr. Manzan, and we're going to be posting it. Both of us are going to be posting it in our Canvas calendar for today. So if you want to focus more just on listening and learning, please do so, because the notes will be provided to you at the end. Okay. We've also changed the homework assignment, because I know some of you have questions about that, as to whether or not what's going on with the homework assignment. So the homework assignment that was due for today, that we gave you yesterday, is now going to be due tomorrow on Wednesday. So you have some time. If you've already done it, you can go back and look at it, change some things around, and then resubmit it. If you haven't done it yet, today you're going to be learning about it, and you can do that then. The homework assignment that we were assigning today will now be assigned tomorrow and will be due on Thursday. Okay. So this homework, we've already changed the due dates in our Canvas calendar. So you can find this assignment in Monday, but the turn-in will be on Wednesday. Okay. All right. So here we go. Now we're going to start at this place that we call, actually, our learning objective for today is that you, by the end of class, should be able to identify the functions of the enzymes used in transcription to build a mature strand of mRNA. Now, you might be thinking to yourself, well, what is that? I don't really know. That's the point of what today is about, is learning that. And we're going to start right here, what we call the central dogma of biology. Now, the central dogma of biology is really the main focus of what's going on in biology. And without this idea, this central focus, there is no diversity of life. That's how important this point is. And that point is simple, is that DNA is going to make RNA, which makes proteins. And that if we break any part of that chain, we actually don't have the diversity of life that we currently have. That DNA has the code, and that code will be copied to make an RNA, and the RNA will be then to make proteins. And remember, proteins are like hair color and eye color and your ability to roll your tongue are all because of proteins. We also are using proteins that we used in DNA replication, like helicase and ligase and DNA polymerase. Remember, they all end in ase. And so we are going to be looking at more proteins that our body is making that we're using in this process. So that's why this whole idea is super important for us, that DNA makes RNA, which makes proteins. Now, today, what we're going to be doing is we're going to be looking at the process called transcription today. The word transcribe means to write down. Okay? So, in transcription, what we're going to do is we're going to then go from DNA to RNA. We're going to write down the code of DNA, and we're going to write it as I was going to screw that one up. I know Miss Ryan would, if she was here, she'd be laughing at me. A message in the RNA. And that message of the RNA, we're going to actually call, very simply, messenger RNA, or mRNA. So today, the whole point of what we're doing, this is day 4, Tata, of the notes, right? Day 4, right there. Okay. So go to your first page, click on day 4. Now, again, what we're doing is we're going to be making DNA into RNA. That's the process of transcription. How we're going to do that is how we did the DNA replication. First, we're going to watch the video about transcription. All I want you to do is watch. Don't worry about the names of things. Just watch it. Then we're going to break it down, and then we're going to watch it again at the end. I'm going to ask you after the video, what was your understanding again, 0 through 5? 0, I don't understand any of it. 5, I can teach it to your parents. I'm going to ask you that question twice, after the video and then after the video. Okay, here we go. Are you ready? Let's do this. So, let's go back to there.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
Okay. All right. So in the chat, after just watching the video, what is your level of understanding of transcription? 0, I don't understand any of it. 5 is I can teach it to my parents. Please put your number in the chat now.
Getting some 2s, 1.5s, a lot of 2s.
Good. Wonderful. Okay.
Okay, good.
Perfect. Now, let's go through this. Now, a couple of things to clarify in the video. The transcription factors, the TF2D, TF2A, all those different things, don't worry about them. All we need to know are the Tata box, transcription factors, RNA polymerase, ATP. That's it. And they're pretty simple things. Now remember, the whole goal is that we're taking DNA and we're going to copy the code. We're going to write the code down into a message of RNA. So that green ribbon-like thing at the end, that was actually the messenger RNA. All right, so let's go through this then. What does this look like? Okay, well, here we go. What is transcription? Transcription is the process of making RNA from a DNA template. Now, again, if you don't want to write these down, don't worry, just sit and listen, right? Listen and think so you can ask questions, because I'll give you all of the notes at the end. So all we're doing is we're making, again, this goes back to here, we're making the RNA from the DNA template, Okay. Now, where does translation occur or transcription occurs? It occurs in the nucleus of the cell. And you might be wondering, well, why does it occur in the nucleus of the cell? The reason being is because DNA can't leave the nucleus, but RNA can leave the nucleus. That's the reason why we actually have to convert the DNA into RNA, is because the DNA, because of its double helical shape, is too big to fit through what we call the nuclear pore or the nuclear hole in the membrane. But RNA, because it's single-stranded, can. And because it's single-stranded, it's small enough to fit through the nuclear pore. And remember, when we were back in the cell unit, proteins are made on the ribosome. So this RNA is going to go to the ribosome so we can make proteins. So we're going to copy the DNA code into a message of RNA that's going to leave the nucleus and go to a ribosome. Now, what is the promoter to start transcription? What's the name of the box to aid in that? Well, the promoter to start transcription was 6 molecules of ATP. ATP, remember, is just energy. And that energy is being needed because of the Tata box. And here's the things I want you to put in the chat. Remember last week I told you to remember the A and the T bond. And it was important for what reason? And it's different. The A and the T has a different bond than the C and the G. Who can tell me in the chat why that is? A and the T bond versus the C and the G bond. The A and the T is? Ooh, lots of thinking going on here. Remember the bonding. Adenine and thymine, very good. They pair together. Cytosine and guanine pair together. What's the difference between how they bond together? Very good. Thank you very much, Julia. Adenine and thymine have a double hydrogen bond, where cytosine and guanine have a triple hydrogen bond. Now remember, 2 bonds are easier to break than 3 bonds. Now, why then is there 6 molecules of ATP needed? We need that 6 molecules of ATP to break the hydrogen bonds. When we break the hydrogen bonds, it opens up the DNA and it creates this bubble. So if you look at my zipper, right, here's the DNA and it opens up, and what it does is it creates this bubble right here. And that we break the bonds at the Tata box. Lots of adenines and thymines helps to create the bubble. And that's the reason why the Tata box is so important for us, is because it's full of double hydrogen bonds instead of triple hydrogen bonds. And if we had triple hydrogen bonds, we'd obviously need more ATP. So we've got some energy that we're going to put in, and that's going to be used to split apart the DNA. The reason why we have these transcription factors is because they give DNA polymerase a place to bind onto or to anchor onto on the DNA. It gives RNA polymerase a starting point. Now, RNA polymerase's job is to add new RNA nucleotide bases complementary to the DNA strand. RNA polymerase was that green blob, and as it went down, it opened up the DNA, and it then added RNA nucleotides complementary to the DNA strand. Now, what does that look like? So if we have DNA and we have RNA, so we've got then, remember, in DNA, the base pairs would be A and T and C and G. our nitrogen bases bond together. And remember, we technically have a double bond there and the triple bond there. So in RNA, what we get then is T will still bond to A bonds with U. There's our new one. This is different. This is called uracil. uricil, uracil. We still get then C and G and G and C. Those still are two the same, but one difference between RNA and DNA, besides RNA being single-stranded and DNA being double-stranded, is that RNA contains uracil and DNA does not. The other thing to remember in RNA is there are no T's. So RNA contains no thiamine. It contains uracil. Sorry, I spelled that wrong. I knew I spelled that wrong. So uracil. Thank you, Mr. Monzon. All right. So RNA polymerase is adding RNA nucleotides, which are these things here. Okay. The last thing then is the role of the terminator. The terminator is going to stop the RNA polymerase from adding more nitrogen bases. That's it. It just stops it. Okay. So once it reaches the terminator, RNA polymerase breaks apart, and then we get this strand of mRNA. Now, so question so far, I want to pause there, and I'm going to count to 60. So if you wanted to write some things down, you can, or put some questions in the chat for me, please. Okay. I'm going to actually count to 60. I'm going to set my timer for 1 minute. Okay. No one's got questions. Okay. 30 seconds. So I'm just going to continue my drawing here. Okay. So here we go. So we now have this strand of mRNA. And remember, it's going to be leaving the nucleus to go to the ribosome to make a protein. Now, when it leaves the nucleus is this really like safe place. And when it leaves the nucleus, it's going to get bombarded. This mRNA strand is going to get bombarded. And if we don't protect the code or the message, right, inside the messenger RNA strand, what happens is that then the message or the code gets changed. If we change the code, we change the protein. If we change the protein, we change its function. So we want to keep this code from without being changed. So we're going to do two things to it. At the 5' end, we're going to add this methylated cap. And at the 3' end, we're going to add this poly A tail. Now the A stands for adenine, just like here, that's adenine. It's the same thing here, adenine. So we're going to add a bunch of A's at the end. Okay. So the reason why we add this cap, and Miss Klepper, if Miss Klepper was here, what she would say is poly A tail has 3 words. On the third end, poly A tail on 3 prime. Okay, so that's one way you can remember it. So all we're going to do is with this cap, with the cap and the tail, that when then the RNA, mRNA leaves the nucleus, it protects the message that's found in between. So that when we get it to the ribosome, that message is intact. It hasn't been changed. It hasn't been lost. And that's the reason why we need to protect it, is because that message is really important. Because without that message, we don't make proteins. That goes back to our whole idea of the central dogma. Without the code of the DNA, we can't make a protein. So that code is really important for us. Okay, so why does the pre-mRNA strand need to be modified? Right, so it can leave the nucleus without the message of the DNA being changed. And what structures are being added? Well, again, the 5' end gets a guanine cap. We actually technically call this a methylated G cap for guanine. And the 3' end gets the poly A tail, 3. Okay. Now that's it. That's all that we need to do. Now there's actually a second part. There's questions 9 and 10. We're going to do those tomorrow. That's where we're going to start tomorrow. So that's it for the learning for today. Okay. What I'd like to do is to go back and watch that video one more time. And again, ask you the questions of 0 through 5, right? 0 means I don't understand anything. And 5 is I understand this well enough to be able to teach my parents about transcription, right? The idea that we're going to be then taking DNA, right? That we're going to be taking a DNA and we're going to be copying down the RNA into a message. We're going to be using these letters, no T's in it, and that we have to put this cap and the tail on it to protect the message in between. Okay, so here we go. Let's watch the video one more time. See where we're at again. Yep. See if this makes sense now from what we just got done talking about.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site.
Remember, don't worry
Other
about
transcription factors, including TF2A
you just need to know
and
transcription
TF2B,
factors.
then attach.
Attach the RNA polymerase.
These complexes prepare the DNA for the successful binding of RNA polymerase.
Or the attachment of RNA polymerase.
Once RNA polymerase is bound, other transcription factors complete the mature transcription complex.
Okay, we're going to start to see now the ATP coming in to
Now,
break
energy
open the
must be
top
added to the
box, the
system
double
for
bonds.
transcription to begin. This energy is provided by the reduction of ATP into ADP and PI.
There, breaks open the top
RNA
box.
polymerase then synthesizes an RNA template from the strand of DNA.
There's your mRNA.
Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
Awesome. Okay. Here's my question for you. Now that we've watched it, broken down the parts, and we've seen it all again, where are you currently at with your understanding of transcription then? Please put it in there 0 through 5.
Seeing more 3s, 3.5, 3.7, through 4s, still a 2.
Awesome.
But it's okay to be at a 2 right now. We're still learning it.
Yeah. Hey, you guys, this is great. Honestly, when we look at the numbers from where you first were, it looks like everybody went up. That's great. Okay. That's perfect. Now, here's the thing. If you still need some help, if there's something that's still kind of like you just don't quite get it, right? You're not 100% sure, come to the office hours, 3 to 4 o'clock. At 3 o'clock in a half hour, come and ask us just a quick question. We'll be more than happy to help you. Okay. So you guys did a great job with this. What I'd like to do now is to kind of go through the homework really quickly with you to kind of highlight some points with you. So if you could take then a moment and open up the homework assignment, the one that was due for today, that's now due tomorrow, Wednesday. It looks like this. It's called DNA Unit Day 4 homework. Why don't you open that up? I'll give you a second here to put that in. We're actually a little bit ahead of schedule because you guys did an amazing job. Super cool. Very proud of you guys, seriously. Okay. Now, you guys should be able to answer questions 1 through 5 now on this first page. I want to do question 6 with you just so that you guys can see this. What we're going to do is we're going to do transcription. We're going to go from DNA to RNA, right? So really what we're doing is we're going from DNA to messenger RNA. So remember, in mRNA, there are no T's. That's an easy way for you to see if what you're doing, okay? So I'm going to do I'm going to do line 6 for you so you can kind of see what's going on here. So remember, we're still giving the complementary RNA nitrogen base to the DNA, okay? So DNA is cytosine, the mRNA is still guanine. DNA is guanine, mRNA is cytosine. Thymine in the DNA is complementary to adenine in mRNA. The adenine in DNA is now complementary to uracil in messenger RNA. Remember, there's no T. There's the key. C is complementary to G. G is complementary to C. Thymine is complementary to adenine. Cytosine is complementary to guanine. Adenine is complementary to uracil. Guanine is complementary to cytosine. There you go. So you now have a template for you to be able to do. So you can do now 6, 7, and 8, 9, and 10. And then down here on this side, you're given the mRNA strand and you're going to work backwards to make the DNA. So remember, you're going to get T's back here. You just have to figure out where the T is. You can use this as your guide. Use this strand as your guide, okay? Now, on the last page here, there we go. Let's write this in for you guys at the same time. Now, how we do this page is this. The DNA template and complementary DNA, these things are really the same thing. Remember, this is DNA. So we're going to complement, oh boy. Complement the DNA. So A goes to T. I'll even throw the double bond in there, and C triple bond to G. So what you see here, right? TTACG on the template strand of DNA. The complementary DNA would be the opposite of that, AATGC. All right. Now the mRNA will be made using the template strand to make the mRNA. Template means it's the I was gonna say template, which doesn't really help because it's the same word in the definition. The template would be the model that we're going to be basing it off of. So as you can see here, when we have T, the opposite for that in RNA would be A, T to A, but now A for the adenine and DNA gets paired to U in the RNA. C would be the opposite with G in RNA, and G, the opposite of that would be C in RNA. The easy way to check this, that you've got your mRNA strand correct, the easiest way to check this strand right here is to look at this line. You'll notice that they're the same thing. It's just there are no T's here. So remember, again, no T's. So you can just check the complementary strand like you've got here, AATGC. In the messenger RNA strand, you have AAU that replaced the thymine GC. So they're the same thing, it's just we took out the T and we replaced it with the U. Okay? So you should be able to then do the rest of those, figuring those out, okay? Now, remember, this assignment is due Wednesday. For those of you who've already done it, I'd go back and look at it again in Notability to see if you've got it all correct, you've done it all correct. And then if you haven't, make your changes and go back and resubmit it. For those of you who haven't done it, you now have tonight to be able to do it and then turn it in tomorrow. If you don't understand this, come to after school today, 3 to 4, or tomorrow morning, 8 to 10, to get help on it. We would love to be able to help you on it then, okay? So the DNA unit day 4 homework, central dogma transcription, is due tomorrow, Wednesday. Tata, you said you get it. That's awesome. Congrats. You guys should feel really good about this stuff. I think one of the things that you kind of go like, you know, but this is really difficult, but then you go down and you start doing it, it makes sense for you. You guys are doing a great job, and I want to make sure that you guys know that. I really am super proud of you in this because this is a difficult thing. And I think you guys are doing an amazing and an awesome job with it. So keep up the good work. Come to after school if you need it. Come tomorrow morning if you need it. Okay. And don't forget, dress up on Friday so that Austin's class can win. Mr. Monzon, what do you got for us? Anything? The attendance is in the chat, ladies and gentlemen.
Yes, Google attendance form and the exit ticket is in. You have everything you need for getting this stuff done now. Looking forward to seeing your grades. Again, you can update this in your submission if you did make a submission early, and that's fine. You can always resubmit as many times as you need to. I got nothing else for you guys. Great job today. Proud of you guys.
Thank you, Tata. You did a great job with that. I'm really proud of you. Nice job. Everybody, thank you very much. Proud of you. Once you're done with your attendance, you guys can go have a great night. Take care. We'll see you tomorrow.
And here it is
I
again.
have a
if
quick
you need it
question.
again. Good job, guys.
How do I get check the comments for the egg lab?
So you're going to go to the assignment, the egg lab assignment.